Ayurvedic herbal medicines are considered as one of the longest traditions for the treatment of disease as well as health maintenance. Of late, the popularity of Ayurveda herbal medicines has shown a significant rise all around the world, which subsequently leads to the demand for one of the most vital aspects of their standardization and evaluation, which is authentication. This aspect ensures that the material is authentic and true, and is therefore crucial since it is not uncommon that they are either accidentally or intentionally substituted and or adulterated with closely related herb species. Oftentimes, unrelated plants with indistinguishable morphological features and similar common names cause the adulteration. Therefore, proper authentication process is compulsory to prevent such issue. As such, macroscopic, microscopic, and chemical evaluation are usually carried out for the standardization of botanical materials. The macroscopic identity will subsequently be compared to standard references in respect of the botanical material's shape, color, size, surface characteristic, texture, odor, fracture characteristic, taste, etc. In contrast, microscopy focuses on approximate microscopic assessment of powdered or broken crude materials. And finally, the material's characteristic chemical pattern will be defined through chemical evaluation. All of the three methods mentioned are beneficial in terms of cost and rapidity for plant identification. However, they are still not without limitations. One of the difficulties is the variation of composition and chemicals quantity of particular plant species at different growth period. This condition is further worsened by the huge number of compounds contained in each herb, which makes it harder to trace and analyze the availability of all compounds of interest. Therefore, it is imperative to explore for more appropriate tools for plant material standardization. In this regard, DNA fingerprinting demonstrated greater role in the authentication of medicinally important plants. DNA, the basic component of all living cells, is known to be more stable and display less variation throughout different seasons and plant growth phase. Alec J. Jeffrey, a Great Britain geneticist, discovered the basic technique of DNA fingerprinting in 1984. In general, DNA fingerprints involve the process of identifying and distinguishing one individual from another at molecular level through a barcode-like patterns. DNA-based pair sequence arrangement determines the plant traits, characteristics, and morphological features. In addition, it is also responsible for the regulation and production of proteins and enzymes in cells.
DNA-based marker. DNA-based marker can be classified into three major classifications. The first one is hybridization marker. It can be divided into 5 types. The first one is RFLP, second is VNTR, probe hybridization with micro and mini satellite, random genomic clone, and also cDNA clone. For the PCR-based marker, it will be divided into 4 major types. The first one is inter-random sequence repeat, second is DNA amplification fingerprinting, the third one is the amplified fragment length polymorphism, and the last one is random amplification polymorphic DNA. And the last classification of the DNA-based marker is called sequence-based. It also divided into 4 major markers. The first one is called simple sequence repeat. The second one is sequence characterized amplified region. The third one is cliff amplified polymorphic sequence, and the finally is called single nucleotide polymorphism SNP. And for the DNA fingerprinting method, it can be divided into several methods. The first one is about the isolation of the DNA from the plant part. Second, quality and quantity check for the isolated DNA. Next is to amplify the specific gene using the PCR, and then respective genotyping, and lastly about the result interpretation and comparison of the result with the suspected herb. The first methodology is about the isolation of DNA from the plant's part. The DNA can be isolated from different types of plant part, which such as leaf, roots, and stem. This DNA can be isolated from this plant's part. And then the second is about the quality and quantity check for the isolated DNA. The DNA, the isolated DNA, should be checked for their purity, and there's a ratio reading that indicate the purity of the DNA. For the 1.8, it shows the highest purity of the DNA, and if the ratio reading is more than 1.8, it show that there's RNA contamination, and if the ratio reading is less than 1.8, it indicate the protein contamination. The third one is the amplification of the specific gene using PCR. PCR can be divided into three major steps. The first one is heat denaturation. The denaturation temperature is 95 degrees Celsius for 30 seconds, and during denaturation, it will denature the double-stranded DNA into two individual strains. And the highest temperature may be appropriate for the guanine G and also cytosine C rich nucleotide. Second is annealing. During annealing, one of the primer will bind to the 5 end of one DNA strand, and another primer will bind to the third end of its complementary strand. And finally, primer extension. In primer extension, the temperature at this stage may be vary depend on the tag DNA polymerase, but at the temperature of 72 degrees Celsius, the nucleotide incorporation vary from 35 to 100 nucleotide per second. And after the application of the specific gene using PCR, this goes for the respective genotyping. In the respective genotyping, there are several methods for the respective genotyping, which are RFLP, ALFP, ISSR, and also RAPD. And finally, after the respective genotyping, it's about the result interpretation and the comparison of the result with the suspected herbs. So this is the result, the PCR result. There's a gel electrophoresis reading for the isolated DNA and the comparison between the suspected herbs, and the result interpretation and matching should be done by comparing the isolated DNA with the suspected herb.
DNA-based marker, utility and usage in Ayurvedic herbal medicine. DNA fingerprinting or DNA profiling is known as a highly specific technique that are able to read and identify different organisms' individual genetic constitution, including plants. This is very useful in differentiating adulterated or mixed herbal medicine, even it may be indistinguishable morphologically or phytochemically. With known molecular markers of target plant, adulterant can be distinguished even in processed product, enabling the authentication of the drugs thanks to DNA specificity. So, many of these techniques has been used for multiple aspects in Ayurvedic herbal medicine, such as in identification, detection of adulterants, differentiation between species, and also identification in compound formulation. For example, in identifying the species, Swatia species, or known as Tira tatitaka, has been identified using RFLP and SCAR markers. Of 483 base pair amplicon marker has been identified to belong to Swatia chiraita. Next, adulterants can be detected using DNA fingerprinting in different samples. In this case, RAPD markers has been utilized to identify adulterants in many species such as Emblica officinalis, Andrographis paniculate, Termina bellerica, and Terminalia cebula. Also, a sample of Bacopa monnieri was able to be distinguished from its adulterants, such as plants from Centella asiatica and Malva rotundifolia, thanks to the development of RAPD-based SCAR marker that identified 589 base pair markers specific to Bacopa monnieri. 。 Other than that, DNA fingerprinting can also be used for species differentiation. For species differentiation, scar marker was used to identify Phyllanthus urinaria by recognizing 319 base pair unique primer to that species. Other example for this case would be in the case of differentiating and authenticating of aerial parts of Texas baccata from other species by using single nucleotide polymorphisms or SNP. Other than that, these methods can also be applied in identifying the formulation in the compound of herbal medicine. For example, compounds contained inside an herbal medicine prescription, Rasayana Churna, has been identified by using RAPD by a primer called OP6. Through this method, the contents of Rasayana are determined as dried fruit of Amblyca officinalis and Tribulus terrestris, and also dried stem of Tinospora cordifolia. In conclusion, it has been proved in multiple publications that DNA fingerprinting has myriads of utility in authenticating medicinal plants. Its ability to differentiate within and between species is very useful, allowing it to detect adulterants as well. This acts a proof that DNA-based tools is the future in pharmacognostic method in assuring quality in both research and industrial usage. This is due to their flexibility and power in detecting adulterants that is very common in herbal medicine and works in any form of drugs, whether processed or not.